Hey everybody, you're watching Class 1 of a new lecture series called Tidybiology, an introduction to biological data science in R. My name is Matt Hershey and I'll be your instructor for the next 5 lectures. The video you're about to watch is a recording of a class that I taught in the fall of 2019. The class is targeted towards graduate students in molecular biology, so several of the comments and references within the class will be targeted to this audience. But importantly, the information and the content related to data science within this course is broadly applicable. We assume no prior knowledge of R, the programming language, or really anything about programming, and really are looking for people who are eager to learn data science. If you haven't yet installed the R or RStudio software, then go ahead and watch the installation video before diving in. If you have, then it's time to learn.
In this first class, what I want to do is set the stage for you. I want to give you a sense of why you're here, other than it seems like R is a good thing to know, or my PI told me that I need to learn R, right? So in a bigger sense of why we're here, and I'm going to take probably 20 minutes or so now and set up the stage and set up the context for what we hope to accomplish and what we want to do. And then we're going to do some code-alongs together. And then every single class from then on is essentially, there will be a little bit of lecture. Today will be the longest, again, probably like 20 minutes or so. And really what we want to do then is work together through some problems, through some data sets. So I sent out the instructions. Hopefully you guys have done that. I also sent out the syllabus. Hopefully you looked at that. If you have questions about those, let's save that to the end so we can really just dive in. So again, welcome. This CMB module is called Tidybiology. You'll see sort of why throughout the course. And really this is an introduction to biological data science in R. So data science is this new and emerging field and concept. In the biological sciences, we've called it computational biology. We've called it bioinformatics. We've called it statistics. We've called it math and modeling, right? And so this concept of data science captures a lot of these ideas. And so this is a course that is specifically targeted and focused for people who are biologically minded. So hopefully that's you all. All right. So I want to start the course by actually telling you a story. So this is a lightly edited, but a real setup of a paper that I came across that's sort of related to my field. But I want to read this for you for just a moment. So the point of the paper was to identify a new enzyme or a new gene that has a potential enzymatic activity. So identification of your favorite gene as a potential YFGase, okay? So so they took a metabolite-centric approach. So because this metabolite is unique, we hypothesized that enzymes with these special domains might have YFGase activity. Okay, that seems fine so far. To identify potential candidates, we first searched the Swiss Prot database using terms YFGase and metabolite as keywords and obtained 31 candidate proteins. All right, so they went to a database and they said, all right, we're interested in something that has this enzymatic activity, so we're going to search through a database to see if anything is named as having this enzymatic activity. All right, that's the first problem. Okay, next, we excluded enzymes expressing yeast because yeast do not express enzymes like this YFG. After these screening steps, we obtained 9 candidates, including all YFG family members. Among this, they expressed one, and we think that this is the most important, so that they went on and they showed that, look at this gene might do this thing, right? So the details aren't important, but what problems do you see with this rationale for a line of investigation? What is the rationale for the line of investigation? I'll give you a hint. It's in yellow. So what did they do? Search a database using the name, right? The name of the thing. Okay, now, is anybody here working in Drosophila? All right, what's your favorite Drosophila gene?
Scarlet.
Scarlet, exactly. What does it do?
When it's mutated, it produces a red-eye phenotype. Otherwise, I think it is involved in pigmentation of eye color.
Okay, so gene involved in pigmentation of eye color, right? Well, why didn't they name it pigmentation of eye color, right? The Drosophila field is sort of famous for having crazy, clever Drosophila gene names. So my favorite Drosophila gene is INDY, I-N-D-Y. It stands for a gene that is called I'm not dead yet. Because when you knock it out, the Drosophila is long-lived. True story, right? So the name of the thing doesn't really tell you anything about the thing. Does anybody know who this is? All right, this is Richard Feynman.
But it was the opposite. My father had taught me, looking at a bird, he says, do you know what that bird is? It's a brown-throated thrush. But in Portuguese, it's a montarapelo. In Italian, a chuterapihita. He says in Chinese, it's a chongonpo. In Japanese, a patata podecha, etc. He says, now you know all the languages you want to know what the name of that bird is. And when you're finished with all that, he says, you'll know absolutely nothing whatever about the bird. You only know about humans in different places and what they call the bird. Now, he says, let's look at the bird and what it's doing. We knew the difference between knowing the name of something and knowing something, which I learned very early.
So Richard Feynman is a famous Nobel laureate physicist, worked on the Manhattan Project, solved some of Einstein's unsolvable problems. We'll just say smart guy. But he highlights here in this interview, there's a difference between knowing something and knowing the name of something. Because I would argue that the goal of science is really to know something, right? That's why you all are in graduate school, is because you want to figure something out. And if you do, they might give you a PhD along the way. But the whole point is to know something, right? And so this is Isaac Newton. And so I quite like this, because there's a humility. He says, I seem to have only been like a boy playing on the seashore and diverting myself now and then, finding a smoother pebble or prettier shell than the ordinary, while the great ocean of truth lay all undiscovered before me, right? That's all of us. So as students, right, of biology or whatever your sort of subspecialty is, like we are trying to know things, not just the names of things. And so how do we do that? Well, of course, we start with the scientific method. So we define a question, typically, right? Then we observe. So we gather information and we think really hard. Then we form a hypothesis, right? And then comes into what you guys do. Test the hypothesis, perform an experiment, collect data, hopefully in a reproducible manner. Analyze the data, right? This is a null significance hypothesis testing, right? So if it weren't true, then I would try to prove that it's not true, right? It's a little convoluted, but that's what we do. And then we ask the question, is there enough evidence to reject the null hypothesis? Okay, so from this, we then interpret the data, draw conclusions, and then we start the whole thing over again, right? And it's a starting point for a new hypothesis. So this is what we do, right? And now you're probably all nodding your heads and saying, why is he teaching me, or why is he going over the scientific method? Well, because I think we are at a point in science, especially the biological sciences, where there's a new sort of method that is usurping this, okay? And what often happens is we often start with screens, right? So the screens are not really involved in the scientific method, right? So, we joke, well, I had no idea what to do, so I did a screen, right? We do some omics. We do some genomics or proteomics, or we throw omics at the problem, right? Anybody done that yet? You guys, yeah, okay. Anyone else? Yeah, be proud, it's fine, right? So we gather data about the system, but we don't need really a directed hypothesis about this, right? And there's several different sort of levels of this: epigenome, transcriptome, proteome, metabolome, microbiome, etc. All right, so now we could potentially put screen in here. This is sort of independent of a hypothesis. We say, okay, well, we want to know what our gene does, right? YFG, your favorite gene. We knock it out, and maybe we do some screens to get some data from which we can form a hypothesis, all right? Because this is the whole idea. We get data, we turn that data into information. That information then turns into knowledge, and hopefully that knowledge we can turn into wisdom, which is sort of actionable, right? That we can do something with the knowledge that we gained. So I want to set the stage here because there's this emerging field called data science. And this is what I sort of started the very first slide saying, is that data science is a type of, it's an activity that we can perform that goes from this data to information that allows us to more quickly or more efficiently turn this into knowledge, right? And so this is where the data science tools come in. And this is my goal of this module for you. I realize we have 6 classes together. That's not a lot of time, especially because the final class is dedicated entirely to your presentations. So the entire first 5 classes is our giving you the tools to take a data set, to turn it into information, and perhaps even knowledge. And then in that 6th class, you will then share that with us, your analysis, what you did. It's a 2 to 3 minute presentation. So we'll go over the details of it a little bit later, but I want to let you know where we're going. This is my goal for you, to give you the tools to take a data set, to put it into an environment that allows you to extract information from it. Okay, so this is where this emerging field of data science. So what exactly is it? All right, well, this was, this field is like maybe 10 minutes old, right? So this is like 2015. So people are still trying to get their minds around what exactly data science is. So there's some math and statistics, right? R, the programming language, is the quintessentially a programming language that statisticians, especially in academia, use. All right, so there's math and statistics, so you need to have some of this. But then also it's messier, right? There's like some hacking skills that go in, right? I'm not a trained computer scientist, not by any means, right? So you say, well, why am I standing up here giving you a series of lectures about data science? Well, because this is not exactly computer science, right? It's perhaps messier, but then also it is a bit like the Wild West now, where a lot of different tools work, and we'll get to the tools in just a minute. But there's also this element of some sort of expertise, right? This expertise then is required for insight, right? So anybody can take data, but to take that data and turn it to knowledge, you need to have some insight to say, well, this doesn't behave as expected, or I looked at the data in this clever and not necessarily intuitive way, and I found these observations, right? So this sort of Venn diagram perhaps is where data science lies, right? So again, not my Venn diagram, but you can see maybe traditional research falls in here, danger zone sort of in here if you don't know enough about the math and statistics, perhaps. Okay. Now, this, of course, has been updated several times. So here's one, right? So you can look. We've got still our hacking skills, our math knowledge, substantive expertise, and then we've got this sort of evil that comes in. Okay? So right here is your thesis advisor. So we've got some expertise, a little bit evil, and some knowledge of math and statistics. So here is your outside committee member, right? So we've got mostly evil, a little bit of math and stats knowledge, not much expertise. Okay, so we could see, though, that this is sort of an amorphous sort of concept, right? And I'll use some of these terms interchangeably, bioinformatics and computational biology and data science, because it's sort of a blend of all of these ideas. Okay, so our overall goal is to generate knowledge, and that's what we're going to focus on today. Okay, so we can find a lot of different surveys and attempts to quantify, which languages are programming languages are going to be the best to use for these types of activities. And so you can see that this is sort of animated change in the graph over time, and you could say, oh well, C# is the most popular, and Java is really important. And you say, well, Python's so hot right now, especially with all the machine learning, and R doesn't even show up on here. It's like all of this sort of tension about which is a language that I should use. And I'll first show you this and say, well, if you take all of these and you compare them to something like Excel, right? Like, who knows how to hack Excel, right? To take a column and to find the mean of a column, and then take that mean and subtract it from another mean. Yes? Okay. Most people are nodding here. All right. So we all can do this in Excel, right? But we also have this sense that you can see the writing is on the wall, that we are not far from a stage where we need to be able to say, these are our data, this is the code, and this is the figures that it generated, right? Because Excel is great, it's quick, it's dirty, but it's terrible because it's quick and it's dirty, right? So we need something better. So which language, right? Which should we use? Well, it doesn't really matter. If I were going to ask any of you, and I'd say, okay, let's say you had a favorite gene, and you wanted to figure out what it did. What approach would you take? Anyone? What approach would you take? You got a list of genes, you did some omics screen, gene at the top is something completely unknown. What would you do? What's that?
Web search.
Okay, web search. What else would you do? Let's say experimentally, what would you do? You really want to figure out what this gene does. What's that?
Delete it.
Delete it. From a cell, from a mouse, from a Drosophila, something like this, and then phenotype the animal. Great, except that doesn't totally tell you what it does. It tells you what's wrong in the absence of it, but I like your thinking. What else?
Overexpress it.
Overexpress it. What would you do when you overexpress it? Like see who it sticks to, look at maybe the cell cycle, see if the cells are growing differently. All right, there's a lot of different things. Well, which one is the best? Yeah. And it depends on the question. And it depends, right? It depends on so many things. So I'll come back to the question. Which is the best programming language that you should use for the task? Depends, right? Like if you're in a Drosophila lab, maybe you should think about making some knockout strains, right? Or if you're in a biochemistry lab, maybe you want to do some enzymology, or you want to see who it sticks to, or right? So it depends on your skill set. It depends on the question. It depends on a lot of these things. That is the sense I want to impart on you all about this debate of which programming language. We will use R exclusively in this module for one simple reason. I know how to teach you R, okay? And some of the activities that I've been able to sort of accomplish have all been in R. Maybe next year I'll stand up here and be teaching Python, maybe not, but it doesn't really matter, right? I want to give you guys tools that allow you to dive into this field. Okay, now, that's where we're going. That's my hope for you. That's the goal. Any questions so far? We're about to get sort of hot and heavy with the R language. Okay, so the R language. R is a language. Who speaks a language other than English? Who speaks a language other than English that maybe they learned in high school but not really well, but you could maybe say hello to somebody on the street in that language? Okay, now a lot more hands. Okay, so R is a language, right? We are going to be learning a new language together. That language is called R. All right, so there's a couple of things that I want to first lay out. So R has values. A value refers to sort of a thing inside of R, okay? That's all I want you to know right now. R also has objects. So an object can be something that stores information, okay? So the value, 22 over 7, okay, that's a value, that's a calculation, that's a number. And in this case, we can assign that, and we use this thing that looks like an arrow pointing to the left. That's supposed to be an arrow pointing to the left, okay? Literally, it's like that chevron key and the dash, okay? And what we can do is we can assign that number or that value to an object, and we store that object and we can use it for later, okay? So a couple of important distinctions about R and this language. So an object is a name without quotes, all right? It can have some numbers in there, okay? But it's a name, but it cannot have quotes, all right? Followed by this thing, it looks like an arrow. And then we have a value, an object, a function result, and so on. So objects can be a lot of different things. You will make a lot of objects. You will probably make more objects than you need. And all of these objects are stored in a place called your environment. And your environment will be very, very messy. And as you get more familiar and more comfortable with R, what will happen is you will decide and you will have a better sense of, do I need to store this intermediate object or not? Can I just continue right along through? Okay, so if we want to put multiple values in an object, we can combine these values with this C parentheses. Okay, so now we have an object. Object is still called X, but we can have three different things, three different values inside of this object. All right, R also has functions. We're going to be doing a lot with functions. Functions, we'll also refer to them as verbs. So they act on objects. They can also act on a value. Okay, so if we want to do something, right? So what do you think round does? It's a function. Rounds digits, exactly. And how many in this case? To 3. And what is it going to do? How many times is it going to round something to 3 in this example? 3 times, right, because we have 3 different values inside of our object. So if we ran all of this, what we would get is something that looks like 3 numbers also in an object, or actually 3 numbers from this object that are rounded. Okay, so again, this is a name without quotes, followed by a parentheses to run the function. Okay, so this is the name of the function. And the parentheses is, we're going to go into this more, but the parentheses then are some of the arguments for the function. So in this case, the thing that we're running it to, and this is an argument that gives some more information about it. Okay, so R is a language. We have values, objects, functions. Okay, so which of these are numbers? We'll walk through this together. Is this a number? Yes or no? Yes. Is this a number? No, why not? Because it has quotes around it. What about this one? No. What about this one? What is it? It's an object. Exactly. So this is a number. These are words. We also refer to words as strings. This, exactly, is an object. Okay? Not an object because it has quotes. Not a number because it has quotes. So quotes is how we define something as a string or a word. Okay. Now, which one of these will work? Suppose that, which of these will work? We'll just call this A, B, C, D. Which ones will work? Will this work? Yes, because we're taking the log, which is a function of a number. Will this work? No, because you can't take the log of a string. Okay? Will this work? No, it won't work because you can't take the log of a string. Will this work? Yes. Why? Right, because this object stores a number and we can take the log of a number. All right, these two. Good. So, more introduction. So data are stored in tables and data frames. Essentially the same thing. There's also a special kind of data frame called a tibble, which is something that you'll learn about a little bit later. But essentially, this is the way data are stored. Like that looks familiar, right? So you all shot your hand up when you said you're familiar with Excel. So just like Excel, we have spreadsheets with rows and columns. This is a data frame with rows and columns. So we have some row numbers, we have some column names, and then we have some values in each of these. Okay, so it turns out, I'll just mention actually, that this data frame is called chromosome. You'll see it again, okay? So from here, we can call a single part of the data frame. One way to do this is we use the data frame name, okay? So this is an object, right? Because we said that an object is just a unquoted name of something. So it turns out that this object's type is a data frame. And we can then call a part of the data frame by using this money sign and ID. If we wanted to call a different part of the data frame, we would just put in a different identifier. So ID is here. Let's see. Nope. ID is here. If we wanted to pull out length, we would just say length in millimeters and so on. Okay. So we can call this a single part of the data frame this way. We can also save it as an object. So we can take this, and now, again, using this left-handed arrow, we can call this IDs or something like that. Okay? Now, IDE stands for an integrated developer environment. So there's a difference between R and RStudio. So R is going to be the underlying engine that is driving all of the computation. Okay, so this is R. As I said before, it's a statistical programming language. Okay, RStudio is an integrated developer environment. There are several different integrated developer environments for several different programming languages. So if any of you have heard of Jupyter or Jupyter Notebooks, that is an integrated developer environment that is oftentimes used with Python, the language Python. So RStudio is a common and popular and intuitive IDE for R, which is what we'll be using for this class. So what you can think about and how to define or how to think about the differences between these is R is the engine, okay? RStudio is the car, right? That allows us, we use the engine to drive the car around, right? But we don't interact with the engine. We interact here. And there's a lot of other things that allow us in RStudio that make it simpler to write code in R and to perform analyses and to carry out projects and so on. Okay, so what I want to do is I'm going to do a short demo. If you want to follow along, you can. So 2 things to mention. If we look at, let's see, RStudio. So you guys should log in here. You should have a workspace called Tidybiology 2019. All right. And then if you want, you should be able to list your projects, right? Because I'm the administrator, I see a lot of your projects. They're all the same, okay? But the one that, if you haven't made that copy yet, you should see learning with my name. So click on that. And that should open up a project. So project, I'll get ahead of myself. Okay, so this is RStudio Cloud's environment. If you look at RStudio, this is RStudio's environment, okay? Other than being in dark mode, they look essentially identical. RStudio Cloud has this menu over here, but you can see they're identical. All right, now this console is R, and we can do things in R. So before we do that, though, let me just give you the short orientation. So you have a project here. So the way that RStudio stores groups different data sets and analyses together are in projects. So if you have a project, it's it's quite intuitive because you all work on projects all the time in the lab. How granular you want your project to be is up to you. If you have one analysis, you could call that a project. But if you have 10 analyses that are all related to trying to do some proteomic analysis or something, that could be a project also. I'll leave it up to you. We have a single project, and that project here is called Learning. In this project, we have, if you click on the Files tab, which is this one right here, we have several different files. The pre-class information, if you just want to look at it, is the paper I sent out, the install instructions I sent out, the pre-class email, and the syllabus. Okay? Then, for each of our 6 classes together, we have a class that's called Class 1, 2, 3, 4, 5, and 6. We are in Class 1 today. This is the introduction. Okay? Help file vignette. We'll get to that in just a moment. And then slides, if you guys didn't see, PDFs of all of the slides that we will be presenting are here. So if you want, you can open up the PDF. You can make notes on it. It's yours to do what you want with. Okay? So if you come to intro and then you open up demo, you'll see a very short demo file. Okay, we'll get to what type of file it is in just a moment, but this is a demo file. All right, now, if down in the console, okay, this console here, this console is the RStudio, is the R console that you would get if you just opened up R. So this is R. So we can do things. We can say, show you. It's loading. All right, so we can do things like 10 times 10. It's 100. This is a fancy calculator, right? So, and then we have 10 times 2, 10 times 3, etc. But we can also do things like C, 1, 2, 3 times 10. And we get 3 numbers, right? 10, 20, 30, right? Because this is basically combines all of these, and we perform an operation to it. In this case, we just multiply, right? Now, we can also assign an object. Oh, I didn't want to do that. I wanted to do this. We can assign this object, and if we type x, x now is just this combined values of 1, 2, and 3, okay? And now we can do things to the object, right? We can multiply it by 10 and so on. But remember, if we go like this, it doesn't work. It says error, x, a non-numeric argument to a binary operator. All right, our first error. You'll see several of these, okay? So what is it saying? Non-numeric argument. What does that mean? Yeah, like x in quotes is not a number, it's not numeric. So you can't do something to it that requires an operation to it that requires a number, right? So, so this is R. You can do all of these things in R. You can save files in R. You can load scripts. You can store objects in your environment and so on. Like this is all absolutely possible. But there's a better way, all right? Because what we want to do is we want to get to the point where we have reproducible analyses, okay? This is RStudio, all right? So what we can do is let's just take x, and we're going to call it Matt, okay? So now you can see in my environment over here, I have a value. This object is x, and it stores, this is a character, Matt, right? Has quotes around it. Now, one of the things that I like to do oftentimes is run this command here. This clears my environment, right? So if I come down here and I type rm list equals ls, Matt goes away. The object Matt goes away. All right. So we have a clean environment. And now what we can do is we can do the exact same operations here where we can take 10 times 10. Okay? We can type them down here. Right? Because again, this console down here is exactly the same as the interface I was just showing you in R. Okay? But this is also then allows us to save some of our steps that we're doing. We'll talk about how to execute things in here in just a moment. Right? But what we can do is we can see that we can do 10 times 10, 1, 2, 3. We can do things like combine these values. We can store them. We can multiply them. And we can also get the same errors. Okay? So we've got a console down here. And we've got a file that we're doing things to here. All right? So this is our integrated developer environment. This is what we're going to be spending most of our time in together. Okay. Questions about that? All right. So we have this, this integrated developer environment. Now what we want to do is we want to spend more time with what I was just showing you. I sort of quickly went through it, but I want to more slowly go through and basically have you guys now do an exercise. And we'll take some time to explain this, but we'll take 5 minutes now. And I want you to go into this the intro folder where we just were. I want you to open up 01-exercise.rmd, read through the file, and do everything it tells you. So we'll take 5 minutes. We'll do that now. Okay? So the R Markdown document is a combination of code, notes, code results, output, all sorts of things. The note here makes me actually remember to tell you that commenting your code is super important, right? So you saw a note probably in there to ask me to remind myself to tell you to comment your code, okay? And it turns out that we want to get you in the habit of writing more notes than you think you need, right? So there's this verbose style of writing code. Mostly, you write these notes so that when you go back and you look, you say, well, what did I do here? And why did I do it? And what was I thinking? And so on. Like, those notes are for you, all right? So you want to be able to have some text, some code, some notes about the code. You could write your notes about the code in text above. That's fine too. But anyhow, so the R Markdown document, this RMD, is what we'll be working with for this entire module. And the power of this is that we get to integrate all of these sorts of things, all of these elements. Okay, so we don't have to store one script here and another script there and some notes about the scripts and all of these things. Like, you can read and follow along and execute the code as you go. All right, so R packages. One of the nice things about the R ecosystem is that there are tons and tons of functions that allow you to do operations and to carry out transformations of your data in ways that you don't have to invent. When we put this all together, we put them together in ways that we call R packages. So there's something we refer to as base R. So base R is when you install R, and whether you're looking at it through the console or whether you're looking at it through the integrated developer environment, there are functions within base R that then allow you to add and subtract and round and do different types of statistical transformations to your data. But let's say you wanted to do something to your data that doesn't exist, like you've got some genomic data or proteomic data or other types of biological data. Well, fortunately, people have developed what we call R packages that have functions, they have data, they have help files, they have vignettes, they have this information that essentially what you can do is then you just pull down this package and you have now all of their functions. So this is great, right? Because now you don't have to invent anything. You can say, well, I want to do this. Is there a chance that somebody else has done that before me? The answer is most likely yes. Okay, so these functions are verbs, okay? This is how you can think about your function. These verbs act on data. So when we talked about the function and how it's written, you say the function name is do this, and what are you doing it to? It's to that. And sometimes they have additional arguments. We do this, to that, using these, right? That's how you can think about it. A function is a verb that acts on data. But functions, or packages, contain things other than functions. They have documentation. They sometimes have data sets. We'll be talking about that in just a moment. But these functions are really what's driving most of these packages. So one of the packages that you'll be using a lot is a package called dplyr. And dplyr is part of a group of packages that's called the tidyverse. And the nice thing about these functions, or these verbs, is they're very intuitive. When we say mutate, it either changes a variable or it adds a new variable to your data frame. We talk about select. We're going to go into these in much more detail, but just to give you a flavor, select. It picks variables based on their names. If you want to select some and not select some others, you can basically choose what's in your data frame here. Filter. Well, this is intuitive, right? We can filter one of our columns of our data frame based on the argument we provide to filter. Summarize. Summarize is a way that we can pass other functions to our data. And arrange changes the order of our rows. So all of these functions are in dplyr along with others. And so all we can need to do is download a dplyr. So where do you go to find these packages? Well, the same place that you went to download R. So it's called CRAN. So CRAN is the host of all of these different packages. And most of the time, you won't go to CRAN to download the data, and you won't even go to CRAN to search for functions. What you'll do is you'll probably go to Google, or you'll probably go to the paper that you read that then they said, oh, we generated this package is on GitHub. We can then, you can go and download that package directly from GitHub, or you can download packages from CRAN. CRAN is a repository for all things R. Okay, there's a lot of packages out there. That's why you're probably not going to go to CRAN to search for these. But what we know and what we can think about is CRAN is sort of this gateway where there's a certain level of development of a package it has to achieve in order to be hosted on CRAN. And you can see that there's a very active R package developing ecosystem. Okay, so now using packages. Because again, we're not going to be using just base R. We're going to be using a series of packages called the tidyverse. So using packages. Now, the way that you do this is I've helped you guys a little bit, okay? But I want to walk you through how to do this because you will have to do this sometime on your own. So step one, you have to install the package. Base R has a function called install package, install.packages, and then you put what you want to install in quotes, all right? Now what this does is it downloads the file to the computer and it installs it on the computer. If your package is on CRAN, install.packages will find it. All right, so that's the simplest way. Now, you do this one time per computer. If you want to update a package, you do the same thing. If your package has been updated, you type install.packages, in this case tidyverse, and it will overwrite your previous package. Simple stuff. Now, what is this tidyverse? I've mentioned it a couple of times. So this is a series of packages for data science. So it's a collection of these packages. They share an underlying philosophy, a grammar, and data structures. And we'll be talking about tidy data a little bit more. But essentially, the packages within the tidyverse allow people who don't know anything about R, like me a few years ago, to really dive in and be able to manipulate data, to extract meaning from data. And so I'm going to be teaching you some of the tools from the tidyverse to jumpstart your own dive into data science. All right, now, the tidyverse contains the following packages. How would you install them? There's two ways that we could do this that you might know. How did we say we install packages? Right. Then we'd say ggplot2, dplyr, told you about that one, tidyr, reader, per, tibble, and so on, right? We would go through all of these. Now, we also could put this as an object. We would say, like, this is a string. We could put all of our strings together, and we could call it like all my packages. We would have an object, and we could say install packages, and we could put our object in. We could do that too. But because the tidyverse is designed to work together, what the tidyverse developers have done, which includes some people at RStudio, is to provide a shortcut for installing, loading these packages that work together. So when we do this, it's the equivalent of doing all of these things. So that's like awesome. Thanks for the help, right? Now, in your RStudio Cloud, this has been done for you, okay? So you do this one time on your computer. So you'll have to do it one time on your computer, install the tidyverse, okay? Now, if you want to use the package, now we could go to step 2, all right? And what we do, oh, that's a typo. What we do is we say library, not in quotes, loads package. We do this one time per R session. So if we want to then use the library, let's say a tidyverse library, or a package that I've developed for this course for you all called Tidybiology, we can load that one time. And we have to load that the first time before being able to use any of the functions in that package. All right. So again, what we can do is we can use the tidyverse to also, as we use it to install the packages, we can use it to load all of the packages. So you'll see in some of the example R Markdown documents that we've written for you is that library tidyverse is all in there. Okay. Now, a good place to put this is in the setup chunk. Okay. And this is what the setup chunk looks like here. It's the very first code chunk. So we just, when you went through that R Markdown example, these code chunks are in gray. Well, this is what it looks like. Sometimes we can name this. If you name it setup, it behaves in a special way. And then we say library tidyverse. So this is the way that we would start our analysis. One thing that I like to do, and I would encourage you all to do, is load all of your packages in the setup chunk. So let's say you load the tidyverse by saying library tidyverse, and then you start working through a project, and you're like, oh, turns out I need this other package. Go back up and load that library right at the beginning, and then go back down to where you were and call whatever functions you need in that package. Okay? So this is what it looks like. The setup label is special. Your R chunks don't need labels. They can have them. We'll work on that later. But I'll just mention that there's, the setup label is special. All right, now, what I want you to do is first go back to exercise 01, or is it the 01 underscore exercise? And I want you to add a setup chunk as shown below and load the tidyverse package. I also want you to run this chunk. So from the last exercise, there's two ways to run it. You can, since it's just a single line, on a Mac, you can hit Command Enter, or you can hit the green square or the green triangle to run that chunk. So I want you to add this setup chunk to 01 exercise that you just did. Then I want you to uncomment and run the final code chunk at the bottom of the file. So go ahead and do that now. It will take about a minute or two. I won't run the timer because I found the noisy one. Okay. We don't have too much time left, so I want to get through just a few more things for you all. Okay. There's something called exploratory data analysis. This is a concept that's taken from the field of statistics, where you get a data set and colloquially you roll around in the data to understand it. So this concept of exploratory data analysis is a central concept to this tidyverse and data science way of thinking, where you import your data, you tidy your data, transform it using some of these verbs that we talked about, visualize, model, transform, visualize, model, and then finally communicate. So this concept of exploratory data analysis is something that will be central to our time here. All right. So there's plenty of functions that we will spend much more time in the next couple of classes going over. But there's one that I want to show you before we leave, and this is called the pipe. And so this is a famous painting that I don't speak French, but this says, this is not a pipe. Loosely translated. Anyone speak French? How's my translation? No? No one speaks French? Okay, you all believe me then. So this is not a pipe, right? Well, why is it not a pipe? Because it's a picture of a pipe, right? It's a painting of a pipe, right? You can't put tobacco in it. You can't smoke it. So anyhow, I actually think it's really appropriate given what we were just talking about, knowing the name of the thing versus the thing itself. Anyhow, this concept of a pipe has been used in a package that was developed by Stefan Brock, which is used heavily in the tidyverse, which is why I want to take a moment to go over it with you now. So the concept here is that anytime you see a pipe, you say and then. Okay? So the pipe is a sequence of functions that are piped together that are sequentially applied to an object. So you can string functions together, and we do this with the pipe. So here, so we wake up. What do we wake up? We wake up ourselves. We put on clothes, and then eat breakfast, and then go where are we going to equals work. Okay? So when you see that, you want to think to yourself, and then. All right. The alternative way to do it, like this isn't like some crazy invention, like holy smokes, we can string functions together. Like this is really common concept in programming. The alternative way to write this in most programming is by nested code. This is what the code would look like. Go, eat, put on, wake up, self, close, breakfast, to equals work. So the reason that I'm teaching you the tidyverse and this and then way of thinking is because this is intuitive. This we can learn, but let's stick with intuitive for now. Okay, so what does this do? Let's say it together, okay? Wake up, self, and then put on clothes, eat breakfast. We don't know. But that's okay, because where are we going to end up? And then? So what will happen oftentimes is you will see code and you will not understand exactly what every single step is. Okay? That's fine. We can read over that. You can go and look up. It turns out this is find my keys, right? You need to find your keys before you go to work, right? But it's okay if you don't know, because we really want to get a sense, okay? Now, what's the difference between here and here? As an object, yeah. So we save this as a morning routine, okay? So the output of this is a morning routine. Okay, now, one other example. What does this do? Here's a data set. So we take a data set, right? It's called chromosome. And then, or what select does? Selects columns, exactly. So we're going to select ID, which is our chromosome ID, variations, and base pairs. And then mutate. It's going to add a new column called %_var. What do you think that is? Yeah, it's variation divided by base. So this is like the number of variations divided by the number of base pairs. So this is going to be our % variation, right? And then we're going to resort this based on the % variation. This little DSC, this is descend, so we can flip it around. We can either sort this ascending or descending. Okay, so and here what we do is we can save this then as a new object. All right, so it turns out this is what it looks like. We'll see this more a little bit later, but what we have here is the chromosome number, the % variation, and we can then extract some information out of it. We can see that the X and Y chromosomes have much less variation than all of the other chromosomes. So this is our variation, % variation over base pairs. So we'll get to the code for this, but this is where we're going. Okay, so in the final, just if you guys can stay for 2 more minutes. So you're going to have a final project. The final project is going to contain 3 documents, all right? It's going to be R Markdown file with your code. It's going to be a knitted report. We'll talk about that. It's going to be an image of an output. What we want to teach you to do is to take a data set, just like this chromosome data set, and we want to have you roll around in it a little bit, do some exploratory data analysis, and generate a plot that is interesting, slightly more interesting than average. That's the goal, okay? And then what you're going to do is you're going to share the code, you're going to share a report, you're going to show the image that you made, and then you're going to give a 2 to 3 minute oral presentation on the final day, where you will present your code, what you did, the output file, describe the observations. I would say, I was surprised to find that X and Y chromosome have much less variation than the rest of the chromosomes. But then what we also want to do is define a strategy to test a hypothesis, right? So it looks like there's this relationship between chromosomal base pair variation and the number of base pairs, and there are a couple of outliers. So in order to test that, I would, right? So these are the tools that we will give to you over the next 5 classes, right? Now, last idea, writing code is not like drawing an owl. I don't know if you guys have seen this sort of meme, right? So it's not like I've got some code and then I've got this like beautiful sort of insight, right? Maybe this is like if it were in Excel or something, right? No, writing code is a stepwise process. If we have an idea of where we're going, then what we can do is we start with a structure and then we build some walls and so on. And maybe we even comment it, right? We say we finished the 2nd floor and then we started on the roof and so on, right? This is how we want to think about it. So there's a few resources I want to point you to, and then there's going to be a small homework exercise. The homework should take 5 to 10 minutes, right? You guys probably have some experiments to do or something today, right? But I want to give you guys homework each time to allow you to continue this process outside of class. Now, if it's taking more time, then reach out for help on Slack. Or if you look at it and within the first minute, there's something that's like majorly unclear, reach out on Slack. Slack is like our virtual ongoing office hours, right? We're on Slack, all of you guys will be on Slack, and we can all help each other, okay? So there are some cheat sheets that are available. You can Google cheat sheets, or if you look within here, here being your RStudio Cloud environment, you go up to Tools. What you can find, you go up to Help. What you can find is a whole bunch of cheat sheets. All right. So cheat sheets are going to be your friend when you're thinking about dplyr, ggplot2, and so on. All right. So that's on the side. What's this?
It's also right there.
Oh, yeah. We also have some over here too. Okay. So thanks. All right. Resources. Okay. Stack Overflow. When you go to Google and you like Google in some term, like how to remove the legend title from ggplot, it will probably bring you to Stack Overflow, which is this community resource of all things programming. And so Python and Java and C++ and so on. So just know that you could go to a resource like this and type in a question and get an answer. There's also another resource, and again, you guys have all the slides for this. This is an introductory book. You can buy the book, or it's free online at that website that will sort of walk you through some of these things. This could be a resource for you now. It will certainly be a resource for you beyond this class. This is sort of a proper introduction to data science. Okay. And then lastly, there's help files. Okay, so the help files are documentation for functions. So go ahead and open up or look in your RStudio Cloud and type in !mean. What does it do? So go to the console, just type that in. If it works, it should give you a help file. So !mean. All right, the help file is over here. Okay, so it's like, okay, arithmetic mean, and gives a description of the function, usage, and so on. You can also search here. Just type in mean, and same thing comes up. I find myself doing this. I will want to look up like mutate. And what I do is I get a lot of different packages that have mutate in them, but I can go to dplyr mutate, and what I get is a help file for dplyrs mutate. Okay, so those are useful. And one of the other things, this is the last thing for the day, one of the other things that you will do is you will create a small help file vignette. Okay, there is a folder called help file vignette in the workspace. And if you open up students.rmd and run all of that code, what you should be able to do is view a data frame called students. Okay, you can do this now if you want. You can do this later today if you want. Okay, so you all know how to run code, run all of the code in our markdown file. And what we can do is if you type view with a capital V, parentheses students, you will see your assigned sort of vignette and the associated function. Okay, so each of you has one vignette and one function that you will prepare. The reason to do this sort of side project is because you will then be familiar with what it takes to create a help file, to create a vignette, and to basically teach other students about a function. At the end of this, all of these functions, we'll put them online, we'll put them in the package, we'll make them available to everybody, and we'll start building this resource that it's like, well, how do I generate a new column in a data frame, right? Well, if you didn't know anything about that, you would probably Google that, or you would search for that, and you would find out, well, the function is mutate, and this is the way that you do it. So you can see your assigned function. You can then open two other R Markdown, knit the code. We showed you how to knit the code. You just hit that little knit button, or we didn't show you, it was in the exercise. And then you can be inspired. So Ali and Akshay each made one of these that you can then sort of use as inspiration for the template that you will run on your own. Okay, so the last thing, which we ran out of time for today, I'm glad we got as far as we did, is to open up the homework, read through the file and the code, and do everything it tells you to do. Okay? Again, 5 minutes, maybe 10.
All right, that wraps up Tidybiology for today. Thank you for watching. And if you have any comments, go ahead, put them below. If you like us, give us a thumbs up. If you want to see more of this, go ahead, hit the subscribe, and you'll see all of the videos that we post about Tidybiology. Thanks, see you again.